Hi guys, so Jason Matthew here, and today we're going to be looking at a very important topic, enzymes. And today will be part 1, and we'll be looking at introduction, alright? So there are going to be some other parts coming up soon. But let's start with these three guys, and in fact before we talk about them, we should talk about Louis Pasteur, brilliant scientist, and he observed that yeast converted sugar to ethanol. Well, he wasn't the first one to observe that, but he said that there was something in the yeast called ferments that was able to convert the sugar into ethanol, and that led to the vitalism view. Now, what he stated was that the yeast cell has to be intact and alive for the conversion to take place. But Edward Buckner was able to dispute that, because he was able to get some yeast extract. So in other words, the cell isn't no longer intact, the yeast isn't alive, it's just what's inside of the yeast. And he got the same reaction. He was able to convert sugar to ethanol. James Sumner was able to isolate and crystallize the first set of enzymes. He looked at urease, and he saw that urease was made up of protein only, and he postulated that all enzymes were made up of protein. And finally, Haldane, he had a view that he hypothesized that enzymes and substrate bind together via weak interactions. And although it was a hypothesis then, we know now that that is exactly what happened. So some real fantastic stuff was happening. Now in today's class, we're going to be defining exactly what an enzyme is. We're going to state how important enzymes are. We're going to look at an energy profile diagram for an enzyme catalyzed reaction and define what activation energy is. We're going to see how we name enzymes, look at the 6 major enzyme classes, look and see what a holoenzyme is, and lastly we're going to compare inorganic catalysts with biological catalysts. So first of all, let's define what an enzyme is, and I kind of use this kind of cool visual aspect of it. So enzymes, first of all, are what we call biological catalysts. So enzymes are biological catalysts. And because it's a catalyst, it will speed up a chemical reaction. Now, how does it speed up a chemical reaction? Well, it provides an alternative pathway with a lower activation energy. So let's bring everything together now. We are saying that enzymes are biological catalysts that speed up a chemical reaction by providing an alternative pathway with a lower activation energy. Alright? And I'm hoping that you have your pen and your paper with you and you're writing on this, so then you can do it yourself. Alright? Now the first thing I said there was that an enzyme is a biological catalyst. It's a biological molecule. So what is it made up of? Well, most enzymes are proteins. In fact, all the enzymes that we're going to be looking at in this course, as well as the metabolism course, all the enzymes will be proteins. But for the sake of completeness, you should also know that some RNA molecules can act as enzymes. In fact, these catalytic RNA molecules are what we call ribozymes. And these ribozymes, they fit the enzymatic criteria in that they are substrate specific, they speed up a reaction rate, and as like any other catalyst, they remain unchanged in the reaction. Alright, so RNA molecules can also act as catalysts, and we call them ribozymes. So not all enzymes are made up of proteins only. Alright, another biological entity will be the antibody. Now, you know antibodies play a very important role in immunology. Well, some antibodies also have catalytic properties, and we call these guys enzymes. So that's something to note. But also you should know that antibodies are proteins, right? So the two major biomolecules that enzymes are made up of will be proteins, which will be most, all right, over 90% of the enzymes, and then you have some that are RNA molecules, which we call ribozymes. All right, so the next thing we was going to show is that, well, how important are enzymes? Well, this reaction kind of sums it up, because this here is C12H2211. That's sucrose, and sucrose is normal table sugar that you have in your kitchen. All right. Now, when sucrose is oxidized, it gives you carbon dioxide and water plus a lot of energy, and that energy is very useful. The cell needs that energy. So when sucrose enters the cell, within seconds, this reaction will take place, and large amounts of energy will be released that the cell can use. But you know that from everyday experience, that table sugar, which is C12H2211, that can stay on the shelf for years. All right, you could have you could take out a spoon of table sugar right now and expose to the atmosphere, and you will not get this reaction. All right, yet we are saying that this reaction takes place within seconds in the cell to give you that useful amount of energy. In fact, this is what we call respiration. All right, and the reason this can be done is because enzymes, they catalyze these biological reactions. So there are hundreds of enzymes in a cell that are catalyzing a lot of metabolic reactions. All the metabolic reactions that occur in a cell are catalyzed by enzymes. All right, and if these enzymes were not present, then these metabolic reactions that are necessary for life will not occur. So in other words, if we had no enzymes, life itself will not be able to occur. That is how important enzymes are. They speed up the reactions that are necessary for life. Alright, so this is an energy profile diagram that demonstrates how an enzyme works. And remember, as I said, it lowers the activation energy. So on the y-axis we have free energy, and you can see in some books they'll just have a symbol G that will represent free energy. And on this x-axis you have reaction progress. You have substrate and product. Now in the bold line here will represent the uncatalyzed reaction, and in the dashed lines here will represent the catalyzed reaction. In other words, it's the same reaction going on between two of them. It's the same substrate giving you the same product, but in this one with the dashed lines you have an enzyme present. So this reaction with the bold line, there is no enzyme present. In this reaction the enzyme is present, but the same amount of substrate is being used, so therefore the same amount of product. Now if you watch something here, is that because it's the same reaction, the same substrate you're using, you see that the substrate for both the uncatalyzed as well as the catalyzed reaction is at the same energy level, and the product will be also at the same energy level. The only thing that is being changed with the enzyme is this part here, and you see clearly from this diagram that the enzyme is lowering the activation energy. Now this delta G double dagger or delta G double cross, according to some other textbooks, this delta G double dagger here represents free energy of activation, or just simply activation energy. And if you look in the uncatalyzed reaction, the free energy of activation, or the activation energy, is this here. And for the catalyzed reaction, in other words, when the enzyme is present, you see the activation energy is much lower. So the enzyme is providing an alternative pathway with a lower activation energy. Therefore, the reaction occurs faster. Alright. The next thing here is the transition state. So you have S double dagger. And we're going to talk a little bit more about the transition state in the next slide. But it's this point here will represent your transition state. Now be careful when you're drawing the diagrams. Always take from the substrate level. So that's why you see this extended line here. Take from the substrate level and go to the highest point in your curve, and that will represent your activation energy. And for the catalyzed reaction, it's always going to be lower than the uncatalyzed reaction. Now the change in free energy from the substrate level to the product level is called your delta G, or in other words, a change in free energy for the reaction. And as you see, the change in free energy for the reaction does not change whether you use an enzyme or not, right? So there's some things to note for the energy profile diagram. So now let's discuss what the transition state is. During a chemical reaction, the structure of the substrate changes into the structure of the product. Alright, so you have substrate going to product. Somewhere in between, some bonds are partly broken while others are partly formed. So what we're saying here is that if you're going from substrate to product, it means that you will have to start breaking down the bonds within the substrate, and then bonds are going to be formed when you form the product. Now this doesn't happen in a stepwise way, meaning that all the bonds in the substrate will be broken first and then all the bonds in the product will be formed. No, it's a more of a gradual transition, meaning that as some bonds are being broken in the substrate, there are bonds being formed in the product. It's a transition thing. Now this transition state is the highest energy arrangement of the atoms that is intermediate in structure between the structure of the reactants and the structure of the products. Now the transition state is not a chemical intermediate. Don't confuse the two. Alright, it is the highest energy arrangement of atoms that is intermediate in structure between the structure of the reactants and the structure of the products. So we're talking a lot about activation energy, but you probably don't exactly know what activation energy is. So let's deal with that now. So activation energy is the minimum amount of energy for the reaction to occur. And as you can see there, I have put this in bold because it's very important that you include this word in your definition for activation energy. It is the minimum amount of energy for the reaction to occur. You must include the word minimum. Now if you look in this diagram here, you see, well, from here to here is the activation energy. And you can see it will take a lot more energy to convert substrate, which is on this side, to product. So it's going to take a lot more energy to do that. So only a few molecules of substrate is going to be converted to product. But if we lower the activation energy, as you can visually see here, it's much easier to convert substrate to product now. It just goes over easier. Alright, so there's something to note, that by lowering the activation energy, it means that more substrate molecules will be converted to product per unit time, because more of them will have the energy to go over. So the reaction occurs faster. So let's recap what we know already. An enzyme allows a reaction to proceed rapidly under conditions prevailing in the cell by providing an alternate reaction pathway with a lower free energy of activation. So we know that. That's what we discussed just now. Let's add to that now. And this is something you may have covered when you did energy of the cell. But just in case, let's say it now. The enzyme, and it's like any other catalyst this will apply to, the enzyme does not, I repeat, it does not change the free energies of the reactants or products, and therefore does not change the equilibrium of the reaction. So let's repeat. The enzyme does not change the free energies of the reactants or products, and therefore does not change the equilibrium of the reaction. Alright? All the enzyme, like any other catalyst, what it does is that it speeds up the chemical reaction, but it does not change the free energies of the reactants or products. Alright? Therefore, the position of equilibrium is not changed. Now, there are three distinctive features of enzymes. One, they have tremendous catalytic power. Secondly, they are very specific. Right? They react with a specific substrate and they have one type of chemical reaction. And thirdly, these enzymes can be regulated. Now today we're going to talk a little bit about the catalytic power of enzymes. And in subsequent podcasts, we're going to look at how enzymes are specific and finally how enzymes are regulated. So the catalytic power of enzymes, and enzymes are fantastic catalysts. There's no man-made catalyst that could touch this. Most enzyme catalyzed reactions are highly efficient, proceeding from 10 to the power 3 to 10 to the power 8 times faster than uncatalyzed reactions. So what that is saying is that enzymes could catalyze a reaction over 1,000,000 times faster than if there was no enzymes present. Typically, each enzyme molecule is capable of transforming 100 to 1,000 substrate molecules into product each second, and that's amazing. And finally, the number of molecules of substrate converted to product per enzyme molecule per second is called the turnover number, or kcat. I'll tell you that that's a favorite question of mine, so please know the definition of kcat. Again, it's the number of molecules of substrate converted to product per enzyme molecule per second. Alright, it's called the turnover number, or kcat. So nomenclature. And nomenclature is just a big word for saying, well, how do we name these enzymes? Well, this is where things get a little confusing, because sometimes we name enzymes based on substrate. So you have lipase and sucrase, and that gives you an idea of what substrate it works on. Lipase will hydrolyze lipids, and sucrase is an enzyme that hydrolyzes sucrose. So again, the substrate for lipase will be lipids, and the substrate for sucrase will be sucrose. And you'll see that both of them end with A's. And that is something, if you're reading somewhere and you see a word ending with A's, A-S-E, that is an enzyme. You can bet some money on that. That is an enzyme. So sometimes they'll be based on substrate, so like lipase and sucrase. Lipase will be hydrolyzing lipids, sucrase will be hydrolyzing sucrose. Some enzymes, their name comes from based on the action that they're performing. So for instance, in this example here, I'm using pyruvate carboxylase. Now with pyruvate carboxylase, that enzyme catalyzes the carboxylation, or the addition of a carboxyl group to pyruvate. So its substrate is pyruvate, and it's adding a carboxyl group to it. So it's a carboxylation reaction. So we call the enzyme that is catalyzing that reaction pyruvate carboxylase. But sometimes the enzyme, we look at the name, and based on the name only, we know it's an enzyme because it ends with A's, but then we have not a clue on what its function is or what substrate it acts on. At least all we know is that it's an enzyme because it ends with A's. Alright, now catalase breaks down hydrogen peroxide. There's no way from that name you could get that, right? Just based on the name only. And then things could get really tricky when you have enzymes such as trypsin and pepsin. There's no way, just based on the name alone, you have said that was an enzyme. First of all, it doesn't end with A's. Secondly, it gives you no idea what substrate is acting on, and there's no way you could say based on the name alone that what function this enzyme is performing. You just can't. So that's where things get really tricky with these names in the past. Alright, so these are the different ways in which we name enzymes. Back in the day, I should say, right? We're coming up to something that is more systematic very soon. But you see here how things could be confusing. If you think that's confusing, look at this. Glyceraldehyde 3-phosphate, triose phosphate, and 3-phosphoglyceraldehyde. Believe it or not, those three names are for the same enzyme. So as I said, things was getting really confusing in the enzyme world. Alright, a lot of names were being thrown out there, and there was no systematic way. So that is when the International Union of Biochemistry and Molecular Biology, the IUBMB, developed a system of nomenclature in which enzymes were divided into 6 major classes. And within each class are subclasses, and under each subclass are subclasses, within which individual enzymes are listed. Now these classes, subclasses, subclasses, right, they were each given a number. So every enzyme has 4 numbers. Alright? and we call that number, those 4 numbers for each enzyme, we call that the enzyme commission number, or just simply the EC number. So this here is the 6 major classes of enzyme. So you have oxidoreductases, transferases, hydrolases, lyases, isomerases, and ligases. These are the 6 major classes of enzymes. Alright, the oxidoreductases, they catalyze oxidation reduction reactions. The transferases, they catalyze the transfer of carbon, nitrogen, or phosphorus containing groups. The hydrolases, they catalyze cleavage of bonds by the addition of water. Alright, so just remember, important here is that you are cleaving bonds, yes, but for it to be a hydrolase, it must do it by adding water. Alright, the reason why I said it, because look at this. Lyases, they cleave carbon-carbon, carbon-sulfur, and certain carbon-nitrogen bonds, right? But they don't use water. Isomerases will convert one isomer to another isomer, basically. And finally, with ligases, they catalyze the formation of bonds, and they do that with coupled with the hydrolysis of high-energy phosphorus, which is normally ATP. So they do their reaction, and they need ATP for the reaction to occur. So that will be ligases. Now, I require that you know the 6 major classes of enzymes, and I strongly recommend that you know it in this particular order. It's going to come in very useful. So please learn these 6 major classes in this particular order. So look at this example here. You have ATP plus D-glucose to give you ADP plus D-glucose 6-phosphate. And the enzyme that catalyzes that reaction is ATP D-glucose 6-phosphotransferase. That's the systematic name. And as you can see from this name, there's no ambiguity anymore. It is very specific for a specific enzyme. Alright? And the classification number for this enzyme, remember, it's 4 digits. It's 2712. Now remember, I told you in the previous slide that when you learn these 6 major classes, that you know it in a particular order. The reason is this. The first digit in all the EC numbers for all the enzymes, that number represents the major class. So 2 is the second major class. So if you remember 2, then you could go back to the slide and let's see what 2 corresponds to. Transferases. It means that this enzyme here belongs to the major class transferase. So it means that you could get any EC number, and based on the first digit of the EC number, you can say what major class that enzyme belongs to. So I might give you in the exam an EC number and say, well, alright, what major class does that enzyme belong to? And you should know it because you know the order. Alright, now here's the thing, it's very difficult to write this in the literature. It's very long. So you see here, these designations can be cumbersome. So in everyday usage, we still use the common names. So like for instance, this enzyme with the EC number 2712 is simply called glucokinase. Alright, so you can still use the common names, but just to know that there's also a systematic way of naming these enzymes. And all enzymes have been given a systematic name as well as the EC number. So there's no ambiguity. So you could always go and refer to those. Right, so this here is just a reminder of the major classes of the enzymes and what type of reaction they catalyze. So you could just look through this for a revision. Alright, now most enzymes, alright, they are only made up of protein, and that's enough to do their catalytic reaction, right? So the protein component is more than enough to do the catalysis. But for some enzymes, they need a little help. So there's a non-protein component that makes the enzyme work, right? So for the enzyme to function properly, it needs that non-protein component. We call that non-protein component cofactors. Now, cofactors can then be further subdivided. If the cofactor is an inorganic ion, for instance, like Zn2+, Mg2+, in other words, zinc ions or Mg or magnesium ions, alright, those are one example of cofactors. But some cofactors could also be organic, and they are frequently derived from vitamins. Now these coenzymes can then be further subdivided. You have coenzymes that transiently associate themselves with the enzyme. In other words, only when the reaction is going to occur will the enzyme bind to the cofactor, right? Well, the coenzyme to have that reaction. We call that co-substrates. So when the coenzyme is just transiently associated with the enzyme, meaning that it is not permanently bound to the protein part of the enzyme, we say it's a co-substrate. But if the coenzyme is permanently associated with the non-protein component of the enzyme, we say that the coenzyme is acting as a prosthetic group. So there's some terms for you all to know. Alright, so apoenzyme plus cofactor will give you the holoenzyme. And the apoenzyme is the inactive protein part. The cofactor is the non-protein part. And when you combine the both of them, apoenzyme plus the cofactor, you get the holoenzyme, which is the active enzyme. This comes a lot in the multiple choice and so on, so please note it. So these are just some common cofactors that exist. So you have, like for instance, copper 2 ions for cytochrome oxidase, magnesium ions, Mg2 plus for hexokinase, alright, zinc 2 plus for alcohol dehydrogenase and so on. As well as for the coenzymes, you have like biotin for transferring carbon dioxide or carboxyl groups. You have pyridoxal phosphate, PLP, which is found in transamination reactions and so on. And when we continue this course, as well as the metabolism course, you're going to come across a lot of more enzymes, and we're going to be looking at their coenzymes, their cofactors, you know. So you're going to see how they operate. Now we're going to compare inorganic catalysts with organic catalysts, with enzymes. And you see here two very common and popular industrial process. They have the Haber process and the contact process. And Haber process is the industrial manufacture of ammonia, and the contact process is the industrial manufacture of sulfuric acid. You see in both reactions here, it requires 450 degrees Celsius for it to occur. In the Haber process, you need as much as 1,000 atmospheres, so that's 1,000 times the normal atmospheric pressure. In this one, you just need 1 atmosphere. The catalyst for the Haber process is finely divided ion catalyst. For the contact process is vanadium 5 oxide or vanadium pentoxide V2O5 catalyst. Alright, and the point I want to make here, because this is not a chemistry course so much, but I just want to show you all that look at these high temperatures and pressure that are needed for these inorganic catalysts to work. They really cannot touch how our enzyme operates. Just imagine our body is filled with hundreds of enzymes, and at no point in time does our body go at those kind of temperatures, 450 degrees. We just don't exist at that temperature, right? So it means that our enzymes, which are catalysts, they work in our body at 35, 37 degrees Celsius. That is amazing, right? And the catalytic power that these enzymes have is over 1,000,000 times. These inorganic catalysts cannot even come close to that. As well as with inorganic catalysts, you know, they have a lot of side reactions and so on. Because our enzymes are so specific, you are always guaranteed the product. It's never going to have any side reaction like that. You get 100% yield of the product. Alright? You don't get that with these inorganic catalysts. Also, these inorganic catalysts, they are poisoned. Our biological catalysts, our enzymes, not so much. Our enzymes can be regulated. These inorganic catalysts cannot be regulated. So enzymes are really fascinating, and you know, the mechanisms behind it is very, very interesting. So in the next class, what we're going to do? Well, we're going to look at how enzymes are specific. So we're going to be focusing on the active side. We're going to be looking at Fisher's lock and key hypothesis, Koshland's induced fit hypothesis, and we're also going to look at the effect of substrate, enzyme concentration, temperature, and pH on the rate of reactions. Alright guys, so as usual, it has been a good one. Thank you for supporting the YouTube channel thus far. Keep on, just subscribing, liking if you like what you saw, then like the podcast. We really appreciate the support. All right, and we have the Facebook page. You are more than welcome to join my students there. Just one thing, if you're joining, please just send a little note that you saw this on the YouTube channel and you want to join, because we really want only biochemistry people or people who are interested in biochemistry and learning biochemistry and sharing biochemistry to really join this Facebook page. It's not just a big lime or fet, right? We want to really share ideas and so on. So please, we look forward to hearing from you all. And if you all have any figures, diagrams, or any kind of worksheets on enzymes that you all would like to share, please send it to me so I can share it with the rest of the students. All right, it's all about learning from everyone. And usual, guys, thank you for listening to the podcast. I really hope you learned something. Please give us feedback, and we'll talk soon. So good luck, guys, and study hard as usual. Take care.